I have the privilege and honor of introducing our third recipient, Victor Velculescu. Victor was born in Romania. At around age 6, his family relocated to Southern California for political reasons, where he grew up just outside of Los Angeles, and then went to Stanford University for his undergrad training and majored in biochemistry. He then enrolled in the MD-PhD program at Hopkins, where he is now a professor of oncology and has rapidly risen through the ranks. So you've heard about his PhD thesis already. He discovered SAGE. And I just want to remind you of the date of that paper, which is 1995. So that is 16 years ago, really the first view of measuring the entire transcriptome, something we take for granted today. But I think that single paper defines a lot of what Victor's about. He has, in my view, a profound vision of the future and can see the application of nucleic acid-based technologies way before anyone else. So he then joined Bert Vogelstein's lab for his postdoctoral training, and as many of you know, has maintained a lab effort both jointly and independently with his mentors at Hopkins over the last decade or so. And I want to reel off a couple of dates of papers that you all know about, but you probably didn't realize when they were published now in 2011. And they're all defining papers that set the mutational landscape of cancer. So the first tyrosine kinome was published in 2003. The first tyrosine phosphatome was published in 2004. The first microRNA-ome was published in 2006. And the first whole exome resequencing project was published in 2006. And I'll remind you that the Cancer Genome Atlas really didn't get its going in any major head of steam until 2006, 2007. So again, an illustration of how Victor's work is ahead of its time. Now, it's not just descriptions of landscapes. I'll also remind you of a paper that we all know about, a little short brevia published in Science in 2004, one figure that describes PIK3CA mutations in human cancer. And I would argue that that single paper has had a bigger impact on drug development in the pharmaceutical industry in terms of the number of PI3 kinase inhibitors that have now advanced into clinical development than any other such paper. In addition to this award today, Victor's received several others, but there's one that caught my attention, and that is Popular Science named him as one of the brilliant 10 young scientists of the year in 2004. So without further ado, we're looking forward to hearing your talk, Victor.
Thanks very much, Charles, for that very generous introduction. It's a real honor to be here today, to be a recipient of this prize, to be in such a great company of previous winners, of the current winners, and you know, a prize that speaks to the legacy and the amount of incredible work that Paul Marks himself has done. I wanted to also thank Craig Thompson, Steve Goff, and members of the selection committee, as well as Delia, who's here today, my three kids, my parents, and other members of my family who've been very supportive of my work throughout the years. I also wanted to thank my mentors, and I wanted to do that with a quote initially from Sir Isaac Newton, which is, "If I've been able to see farther, it's because I've been standing on the shoulders of giants." And now there's a more recent variation of this quote, probably by a scientist, which is that if I have not been able to see very far, it is because giants have been standing on my shoulders. And in my case, I've been fortunate to have giants of the former kind as my mentors at Hopkins. You've heard Ken Kinsler, my PhD mentor, Bert Vogelstein, my postdoctoral mentor, and a number of other faculty at Hopkins that have inspired me over the years, including Victor McCusick, who's passed away, Steve Balin, and Tom Kelly, who's here today. So what I wanted to do today was to share with you some of the more, you've heard about actually some of the history of cancer genomics in terms of the work that I've done and we've done in terms of looking at individual genes, then looking at gene families, and with the advent of the Human Genome Project and new technologies, it suddenly became clear that your ambitions can really be much larger than looking at individual genes. And so what I wanted to do today was to share with you the efforts that started back in about 2004 and to the present, looking at a number of different tumor types and really performing analyses that have allowed us to look at all of the protein coding genes of these different tumor types. And these have included colorectal cancer, breast cancer, pancreatic cancer, glioblastoma multiforme, medulloblastoma, pancreatic neuroendocrine cancers, and ovarian clear cell carcinomas, as well as head and neck cancers. And these have initially been performed by analyses of Sanger sequencing. You can see up to about here where the amount of sequence that's obtained is large, but is not yet huge. And now with next generation sequencing approaches, the amount of data is really tremendous. Identifying through these various studies anywhere from over 150 to over 1,500 different somatic mutations in over 150 to over 1,000 different genes. These studies really provided for the first time a global picture of the number of alterations that occur inside a human cancer, in a way providing a denominator for the number and types of changes that can actually be drivers in human cancer. They also showed us what the landscape of these alterations are and identified for us new driver genes and pathways, as well as new avenues for diagnostic and therapeutic intervention. And so what I wanted to do today is to share with you an example of how one takes such global data and one can focus in and identify driver genes and pathways. And I want to do this in the tumor type glioblastoma multiforme, where, as you've seen in the previous slide, we performed a whole exome analysis of all of the protein coding genes. But in addition to the sequencing analysis, where we looked for small sequence changes, we also performed an analysis of copy number changes, including dramatic amplifications, as well as homozygous deletions. And if you combine all of these alterations together, you can identify genes in glioblastoma multiforme that are affected solely by copy number changes. This is CDKN2A or P16, which is deleted in about 50% of cases. You can identify some genes like EGFR, which are both activated by point mutations as well as amplification. And finally, there are some genes like PIK3CA, which you've heard about, which is affected solely by point mutations. And I think these examples are important because they show you the value of performing a comprehensive analysis of any cancer genome, of looking not only at sequence changes, but really at copy number changes as well. Had you only done sequence analyses, you would have missed the gene at the top of this list, affected in 50% of tumors. Had you just done these changes, you would have missed genes such as EGFR or PIK3CA, which are greatly and importantly affected by sequence changes alone. So this is, in a way, the way in which you can filter these data and identify those genes which are affected by these multitude of changes. And you can have all sorts of statistical analyses that tell you that the frequency and type of alteration that you see is above the background changes that are typical there. Now, what is unfortunate about this particular study and many other cancer genome analyses that you'll see is that many of the genes identified, in fact, almost all of these, were previously known to be involved in glioblastoma. Now, we know the frequency of their mutation in these tumors more accurately, and we have a better understanding of the type of mutations, but really many of them were previously known to be involved in glioblastoma. However, there was one exception, and it's this gene here, IDH1, which was initially found to be mutated in 5 of 22 tumors in the discovery screen, and ultimately in another 7 tumors in about 80 that were subsequently analyzed. So mutated in about 11% of GBM, and certainly within the top 10 of this particular list. Now, this gene had never been previously implicated in not only GBMs, but in any other tumor type, and not on anybody's list. And at the time, people were making all sorts of favorite lists of cancer genes. And so that was one surprise. The second surprise was that we found that the mutations in IDH were in about 5% of primary GBM. But if you look at secondary GBM, the mutation frequency is much higher. It's over 80%. And if you look at earlier stage gliomas, astrocytomas, oligodendrogliomas, oligoastrocytomas, and others, you see that the mutation frequency is anywhere from 70 to about 100%. In very early stage gliomas, the frequency is almost zero. In other brain cancers, such as medulloblastoma, there were no mutations of this. And in a very large collection of epithelial tumors at the time, no mutations of the IDH gene were found. Now, since then, there's been nice work showing mutations of IDH genes in certain leukemias, as well as in a subset of other adult cancers. But this was clearly yet another surprise. I mean, this is a gene that had not been previously implicated, at the time in any tumor type, and now is a major player in gliomas. So that was unexpected. And the third unexpected aspect about this particular gene was that the mutations at the time all were found to occur in one location, arginine 132, which is this yellow residue here. In IDH2, a related member of this gene family, the mutations occur at arginine 172, the cognate or analogous location, which falls right within the active site of IDH. Now, IDH is an enzyme that converts isocitrate to alpha-ketoglutarate. It's a metabolic enzyme. And you can see here that this particular residue forms hydrogen bonds with isocitrate, and here you have NADP, a cofactor. And for a while, there was a mystery as to what the function of this mutation was, because biochemically, the initial results were that the mutation killed the enzymatic function of converting isocitrate to alpha-ketoglutarate. But on the other hand, the genetic analyses showed heterozygous mutations that all occurred at a single position, which is very typical of an activating oncogenic alteration, like PIK3CA or BRAF or others. And so it's suggesting it's an activating mutation as opposed to an inactivating mutation. Well, more recent work done by Agios and Craig Thompson and others has shown that actually what's going on is that the mutation leads to a new enzymatic function for isocitrate dehydrogenase, whereby the substrate is now alpha-ketoglutarate and it's converted to 2-hydroxyglutarate. And this now provides a very interesting view into a new circuitry, really a rewiring of the metabolism that occurs within these cancer cells. And now for a long time, some people have been telling us about the importance of metabolism in cancer with Otto Warburg and Craig Thompson and Chi Dang and others. And now I think you have a very nice connection between the genetics of a particular gene and the metabolism that has gone awry inside cancer. So much work remains to be done understanding what the function of this mutation is in cancer cells, whether the rewired metabolic circuitry is still important at the time we identify cancers with these mutations, and how this potentially could be useful therapeutically given the new enzymatic function that occurs right within the active site. Regardless of the work that remains to be done molecularly, there's already a clear clinical use of this genetic alteration. The mutation identifies two groups of individuals that have gliomas, and those that have a mutation typically are younger. 50% of the patients between 20 and 40 have a mutation in IDH, while those that do not typically are much older. These are, as I mentioned, typically primary GBM, and these develop rapidly, while the mutations in IDH occur in secondary GBM. These are associated with frequent mutations of other tumor genes, while these are infrequently mutated. And importantly, those that have a mutation in IDH actually do much better clinically than those that are wild type for IDH1. Now, it'll be very interesting, as I mentioned, to see whether this particular genetic phenotype can be helpful for both existing therapies as well as defining new therapies going forward for IDH. And so I wanted to give you this example to show you how these large-scale genomic analyses can be used to identify specific genes, and of those genes, how you can actually identify new genes not previously implicated in cancer, and how those genes could be clinically useful. And this is a very fortunate event. Usually it is much more challenging to make sense of the data that you get in these types of analyses. The number and type of alterations that you get is really quite vast. The frequency of mutations is low. And one needs to think about interpreting these data in a different way. And so one way to do that is to think of these alterations not occurring in specific genes, but rather in underlying molecular processes or pathways that are important in tumor genesis. And so in pancreatic cancer, for example, we integrated the data that we found in a number of different genes with existing databases of gene pathways and molecular groups, and identified 12 core pathways that underlie pancreatic tumor genesis, including TGF-beta signaling, JNK signaling, other signaling pathways, control of cell cycle progression, apoptosis, DNA damage control, invasion, cell adhesion, and RAS signaling. And what's interesting about these canonical pathways is you can actually drill down into each individual patient and identify, these are two specific pancreatic cancer cases, and identify the genes that are actually altered. SMAD4 is, for example, the gene of the TGF-beta pathway that's affected in this tumor, while in this case it's BMPR2. Interestingly, you can also look at gene expression patterns of these particular canonical pathways and find a dysregulation that is way above and way beyond the typical amount of dysregulation that you see in pathways in this tumor type. You can begin to make sense of the data in this way, but I must say it's still quite challenging because the databases that we have of how we understand molecular processes and pathways are oftentimes based on normal cells or model organisms. And really understanding what's different about these pathways in cancer is a challenge and is work that's ongoing. I wanted to share with you a recent pathway that came to light in the analysis of the recent medulloblastoma genome study. This is the list of the top 10 genes. And of these, many of them, again, were known, but the three that were not, MLL2, MLL3, and SMARCA4, and you actually heard about MLL earlier today, all actually belong to a similar pathway. And the recurring theme that began with MLL and now is occurring in a number of different tumor types and with a number of different types of alterations, not only gene fusions, but point mutations, typically inactivating mutations, are alterations in chromatin modifying genes. And examples include ARID1A in ovarian clear cell cancer, endometrioid carcinomas. These are, again, typically inactivating. This is a member of the SWI/SNF chromatin remodeling complex, MLL2 and 3 in medulloblastomas, but also in retrospect, looking back, it's also in pancreatic cancers, again, inactivating mutations. These are histone methyltransferases, as you heard about, DAX and ATRX. In pancreatic neuroendocrine tumors, inactivating mutations, members of the chromatin remodeling complex, SWI/SNF. And interestingly, these genes have an important prognostic indication similar to IDH1, where those that are mutated actually do better than those with wild-type mutations. And there's many others. And what's interesting now is we also have a link, again, examples in a variety of different tumor types, different genes, again, remodeling as well as chromatin modification. And a new link now, I think, between cancer genetics and epigenetics. And clearly, these genes, which affect a lot of different aspects of chromatin structure, will have a number of different epigenetic changes that we've been seeing now for many years, but perhaps now can recognize that there's a genetic component underlying these epigenetic changes. Understanding the downstream epigenetic changes from many of these different genes, I think, will be crucial in coming up with new therapies as we go forward in these tumor types. So I've given you now an example of how you can move from an individual gene to actual gene pathways and making sense of some of this information. But really, this is important for cancer biology, but how do we actually take this information and use it to help patients? And I think this is a very exciting time to be thinking about these sorts of questions because we're, I think, at a paradigm shift in the way cancer is going to be treated. And it's because we now can benefit from the history of cancer genetics for the several decades where we've learned so much about the basis of human cancer, but also new technologies that allow us to look at cancers in a very facile and rapid way. And these together, I think, are going to allow us to look at individualized analyses for both cancer diagnosis and therapy. And I think this is going to be crucial because what we're learning more and more is that there's a great amount of genetic complexity in each individual cancer patient. And this is just an example of two individual colorectal cancer patients whose mutations are laid out on what we call the genetic landscape of colorectal cancer, where the red peaks are gene mountains that are frequently mutated and the smaller red hills are infrequently mutated genes. And they're located on the genome here, starting with chromosome 1 all the way to chromosome X. And each dot represents a mutation of either the first or second patient. And what you can see here is that some of the mutations affect specific genes, while others are located in this green area, which are presumably passenger mutations, tumors or mutations that are not important in this particular tumor type. But interestingly, if you compare these two tumors, you find only 3 genes that share mutations, 2 of the gene mountains and 1 actually that is mutated out here in the gene passengers, perhaps a gene that is thought to be a passenger, but with additional information may turn out to be a driver gene as well. This difference in underlying genetic changes, I think, provides insight into what people have seen in the clinic for such a long time, where you have patients and they might be of the same age, the tumors might look at the same type histologically, they might be from the same part of the body, but when they are treated, they actually respond very differently to a particular therapy and their outcomes are very, very different. And understanding this genetic complexity, I think, will provide insight into those individuals. Now, how can we use this genetic information? I think there's at least two ways to do that. One is to think about using the specific mutations as targets for therapeutic intervention. And we already have a number of examples of oncogenic addiction or oncogenic dependence, as Harold Varmus has called it. And in these cases, and you can see them most clearly, I think, with these examples in the clinic, where you have a particular drug, they work in a particular tumor type, and they work typically best when the target gene is mutated, BCR-ABL translocation, HER2 amplification, or EGFR mutation. And although unfortunate, these genes sometimes, these drugs sometimes lead to resistance and are not fully efficacious for patients, the resistance to these particular therapies is often because of additional alterations that occur within these individual genes or pathways, either alterations within BCR-ABL, alterations downstream of HER2/neu, or alterations downstream of many of the genes involved in the EGFR pathway. And this, I think, provides very important further evidence that, in fact, targeted therapies will work. It's just a matter of overcoming this resistance and perhaps hitting sufficient enough of these individual targets to overcome the resistance. There's many other targets that are now becoming available that you all know about. And interestingly, the number of genes that are being identified as being mutated in cancer is growing exponentially. And although not quite as dramatically, the number of new targeted compounds is also growing at a very high clip. So I think this way of thinking about what mutations are in an individual cancer patient and using that information to target one and hopefully more of their particular mutated genes or pathways will become crucial as we go forward. A second way in which I think this genetic information can be useful is to think about the fact that as cancers grow and invade, they shut off both cells as well as free DNA into both the blood as well as other bodily fluids, such as the lumen of the colon. And the DNA material from these cells contains genetic alterations, and these, as we know, are completely specific to the cancer itself. And so they would actually provide an incredibly useful biomarker and highly specific biomarker for this disease. The challenge is being able to distinguish those few circulating tumor DNAs that contain alterations from the vast amount of normal DNA that is present typically within such bodily fluids. And so to overcome this, we developed a new approach called PARE, personalized analysis of rearranged ins, where resected tumors are analyzed using next-generation sequencing approaches. And this is paired-end sequencing, where you obtain two sequences from every position in the genome. These are typically a certain distance apart, and you can lay these down on each chromosome, as we've done here. But at some points, you find that these pairs of sequences are either too far apart, too close, their orientation's inverted, or in fact, they're from totally different chromosomes. And this is indicative of a rearrangement. And now we've known that rearrangements are present in cancer for a long time, from the work of Boveri and others. It's clear that rearrangements have been there. But now, for the first time, perhaps, we have new technologies that allow us to identify these rearrangement junctions very precisely and to design PCR primers that span the rearrangement junction as a test for the rearrangement in the blood or other bodily fluids. And you can use this personalized test to look at levels of the rearranged DNA over time during therapy. And so this sounds very nice in principle. How well does it actually work? Well, we've looked at, in the initial pilot analysis, we looked at several colorectal and breast cancers and identified these rearrangements. We've now done it in a number of other tumor types and have found rearrangements in all cases analyzed. And so this is a very promising aspect to this approach, because it suggests this is really a universal feature of all cancers, and we can find these rearrangements. How well can you actually detect them in the plasma? That's actually the important question. And it turns out, of all the subset of cases where we've had plasma that we could actually look for these rearrangements in the blood, we've been able to find the rearrangements. In a subset of these, we've actually sequenced the PCR product from the individual patients found in the plasma and found that rearrangement junction is identical to the sequence in the tumor itself, really demonstrating that the rearranged DNA is coming from the actual tumor. So how does one go about using this? I think there's two ways in which this can be useful for patients. One way is to actually monitor disease over time, to provide a molecular readout of tumor burden. And this is an example in one of the colorectal cancer cases where we had serial plasma samples where we could look at the fraction of this rearrangement in the plasma. And you can see here after the initial primary surgery, the level went down, then came back up, and then over a much longer period of time of chemotherapy, as well as additional surgery, removal of a portion of the liver, the levels went way down. Unfortunately, they did not get down to 0, and this was consistent with a known lesion, metastatic lesion that remained in the liver. I think this will be very important in using new therapies and in trying to determine whether patients are having molecular responses to new therapies, to be able to do this in real time. This has, I think, been one of the benefits, for example, in the BCR-ABL field, where they've had a molecular marker like this for some time, as well as in other fields where measuring in the blood the response to therapies has been useful. Another way in which this type of approach can be useful is to answer the question of whether a patient still has residual disease after surgery. A typical question a patient will have of their surgeon is whether or not they've been cured. And unfortunately, nobody can answer that. And so patients are typically treated uniformly. Early stage patients typically don't receive adjuvant chemotherapy. Late stage patients typically do receive chemotherapy, although a fraction of the former are not cured and a fraction of the later are cured solely by surgery alone. And so knowing whether a patient has been cured by surgery and whether there is residual disease or not would allow one to either spare the side effects of chemotherapy or have more intensive chemotherapy if needed. And this is an example of using point mutations in the plasma, work by Frank Deal et al. At Hopkins, a different type of mutation, sequence alterations to look at residual disease after surgery of colorectal cancers. And when no mutation is detected in the plasma, the patients do incredibly well. When mutations are detected in the plasma, the patient succumbs to recurrence and has a very poor outcome. So I've given you some examples of how one can perform these large-scale studies, of the type of data that one finds, of the individual genes that could be clinically useful, and of examples of how I think this will be useful going forward. But that said, there are many challenges, I think, that we all, as a community, face. One is that I think these types of approaches would be much better improved by doing these types of analyses comprehensively, where not only sequence analyses, but copy number and rearrangement analyses can be done for individual, on the same individual patient samples. It used to be that the hard part was finding these genes, but now, and studying what they do was easy, but now the tables have turned. It's very easy to find mutated genes. What many of these do is still very unclear, and even in the case of IDH, a really well-studied now enzyme, very dynamically moving field, there's still a lot to be learned. So I think these will be very crucial going forward for a number of different tumor types. And importantly, as I've been saying through these examples today, I think there's a lot of work to be done to actually make this type of work, which we've been able to do it in the bench for such a long time, actually useful for patients. And it's not trivial, and it requires a lot of thought as to how to actually practically implement it and how to make use of it. I think it'll obviously be useful for correlating these mutation profiles with clinical data as we get more and more information from patients. There are a number of trials that are looking at mutation data and correlating it with outcomes, but there's new trials now that I think are beginning to consider using mutation profiles to actually determine intervention, and I think that's very exciting. With time, I hope this type of information will be useful for therapeutic decision-making and therapeutic targeting, and for disease detection and monitoring. And again, doing this type of work, but integrating it into clinical care will require all the efforts of the entire field going forward. And although we're not there yet, I think in the near future we'll see situations where patients can come in, hopefully their disease can be diagnosed through a blood-based, non-invasive test, and based on the mutation spectra that they have, they'll receive a specific or cocktail of chemotherapy that'll be designed just for that particular set of alterations. So I wanted to acknowledge all the folks that made this work possible. The trainees, Sean Jones, Tobias Schoblom, Laura Wood, and Will Parsons on a lot of the early genome studies, Rebecca Leary and Mark Sawson on this recent and unpublished PARE work, my colleagues Bert Vogelstein, Ken Kinsler, Nick Papadopoulos, Luis Diaz, other individuals that have helped at Hopkins and at other institutions, as well as a number of collaborators from the various genome projects that we've done to date. Thank you very much.
So we have time for a couple of questions. If you have any questions, please raise your hand. And we'll Victor, could you comment on the dynamic range of detecting circulating DNA? I mean, I can imagine, you know, if you're after surgery, if it's negative, someone's going to say, well, how confident are you that it's negative? That kind of question.
Yeah, these are exactly the type of questions that we need to address going forward. But in general, it's thought that there are several thousand DNA molecules per milliliter per milliliter of blood. But that's circulating DNA. We've done some initial analyses comparing the circulating tumor DNA fraction compared to circulating tumor cells, and it seems that the DNA is higher in the cell-free portion. But out of several thousand, it could be that your level is lower than that. And so the method that I've shown you here, PARE, for example, has a dynamic range of 1 in 500,000. And it's probably even higher than that because you're looking for rearrangements. And we simply stopped at 1 in 500,000. But to get 500,000 DNA molecules, you'd have to take a lot of blood from a particular patient, which you just can't do. So there have to be creative ways of thinking, you know, in larger volumes of blood, how much one needs to do. And practically, what is the correlation between detection in the blood as corresponding to tumor burden? And there's studies now that are looking at earlier stages of tumor progression, recurrence, minimal residual disease, and all these things I think will sort themselves out in the years to come.
Of the speakers for wonderful presentations, and in addition we have a reception going on across the street where you can meet them individually and congratulate them in person. We thank you all for coming, and let's